So today we're going to talk a little bit about advances in genetics. And basically what this means is that we are going to choose organisms that have desired traits, and we're going to work to develop those traits to get what we want. So we're gonna answer questions like, how does a liger happen, and why are labradoodles so popular? So that all comes about by selective breeding, where you select organisms with the desired traits that you want, you mate them, and now they're parents of the next generation, and that keeps going and going and going. So, for example, what we normally think of as corn, if we were to trace this back, corn's ancestor would not really look like corn. It doesn't look like anything edible at all. But what scientists were able to do is that they selected the traits that they wanted, such as those really, really fat kernels that in those plants that had a lot of those fat kernels. They mated them on purpose so that their offspring would have those fat kernels and a lot of them. And it kept going and going. And then we chose the ones that were the straightest to give us this nice, pleasing yellow color with lots of kernels on the stalk. And all of this was done just by selecting the traits that we wanted to become parents of the next generation. Now, there's a couple types of selective breeding. The corn is an example of inbreeding, where you have two individuals that have similar characteristics. So you take two corn stalks and you mate them to get more corn stalks. Another example of inbreeding would be racehorses. So they might choose the fastest horses, a male and a female, and mate them together in hopes that the offspring would also be very, very fast and good for racing. Another type is hybridization. So when you cross two genetically different individuals, but the problem with this is that those hybrid animals cannot reproduce. They're completely sterile. And that has to do with the number of chromosomes that they have, because one is going to be left without a pair, and therefore they can't mate and they can't reproduce. So this would be an example of the liger. So when you cross a lion and a tiger, you get a liger. But a liger cannot mate. Another example of this would be when you mix a horse and a zebra, or a zebra and a donkey. The reason why you might do this is because the zebra's genes are resistant to some of the diseases that are prevalent in horses and donkeys. Now, this is a map of the human genome. So you can see that there are 23 pairs of chromosomes. And back in about 2003, we had a really good idea of what the human genome looked like. So we were able to map out all of the base pairs in sequence. So we basically sequenced all of those base pairs, all 3 billion base pairs. We mapped them out, and we know pretty well what base pairs code for what proteins, and basically what traits come about because of those proteins. And that's led to some really fascinating research in the field of genetic engineering. You guys did a little bit of research about GMOs, about genetically modified organisms, where they can take a gene from one thing, splice it, put it into a bacteria, insert that bacteria into the corn or into the tomato or even into the human, and it will reproduce, and then that gene will code for the protein that it needs. So for example, corn that is resistant to herbicides, which means you can spray herbicides on the corn and it will kill all of the weeds that are in direct competition with the corn. And so the corn's going to get all the water, it's going to get all the nutrients, it's going to get all the sunlight because it doesn't have any competition from the weeds. You can also grow tomatoes that can survive in colder temperatures. This also widens the area that you can grow tomatoes in so that the tomato production can be larger. The first human to receive a gene therapy treatment was actually a 4-year-old girl with a very severe immune deficiency disease. And this disease was caused by a faulty gene that fails to produce a vital enzyme. So what they did in the gene therapy procedure was they actually extracted some of her white blood cells, and then they exposed them to genetically engineered virus that still carried some of the normal gene that wasn't functioning correctly in the girl. So the virus invaded her white blood cells, and now these blood cells were transferred back into the girl. So they had that virus had the gene that she was not able to produce. So once back inside the girl's bloodstream, the cells began reproducing the proper enzyme. Now, she still needs some follow up work, and they're still doing some research on her, but basically, she's living a pretty normal life. But the problem with gene therapy is that sometimes when you inject the gene into the virus and then the virus into the person might actually, or that virus might actually attack the wrong genes in the person. And when that happens, it can cause things like tumors to grow and cancer to develop. So while this is still really cutting edge research, some people are going so far as to say that, oh, this is going to cure all the diseases in the world. We're still kind of a long way off from that, but it does give us some promising hope. If you want to check out more about gene therapy and genetic engineering, go back to the Canvas page because I've put some other videos there. Thanks.